Hi everyone, my name is Miriam, and the title of my talk is Recovery of an Ancient Salmonella enterica Genome from a Colonial Individual from Mexico City. To start with some background, Salmonella enterica is a gram-negative intracellular pathogen, and the serotypes typhi and paratyphi A, B, and C are the only ones that infect human hosts. These serotypes cause typhoid and paratyphoid fever, respectively. And as you can see in this image, the infection process begins after ingestion of food or beverage contaminated with salmonella, and through gaining access to the gut lumen, the bacteria disseminate systemically via the lymphatic system, causing perforation and profuse hemorrhage of ileal ulcers. Of note, paratyphoid fever has been less studied and less identified compared to typhoid fever. In fact, an estimated 5 million cases of paratyphoid fever occurred worldwide each year. And as you can see in this map, the highest number of deaths are registered in South Asia. One of the causative agents of paratyphoid fever, which is paratyphoid C, is rarely reported in present-day population. Of interest, this pathogen has been identified in ancient human population through the analysis of ancient DNA. 3 studies to date has identified this pathogen in archaeological samples from Norway, Russia, Italy, Turkey, and from South Mexico. In particular, this last study that analyzed samples from Tepozcolula Yocunda in Oaxaca suggested that paratype C is one of the causative agents of a deadly epidemic outbreak occurred during the colonial period in this particular region called Cocoliskli. Cocoliskli is a deadly disease of unknown cause, which was characterized to cause an intense bleeding and a high fever. To get insight into human infection diseases during the colonial period, we analyzed 7 individuals from Temple of Immaculate Conception dated to the colonial period. According to the archaeological data, this site is associated with the presence of infection diseases. So we extracted the ancient DNA, built sequencing libraries, and generated low-depth shotgun sequencing data from teeth. We use Kraken to identify a pathogen, and then we evaluated the amination patterns to assess the authenticity of the ancient DNA, as you can see in this graph. As the ancient DNA contains a large fraction of environmental DNA, we use a capture enrichment strategy that makes use of biotinolate RNA baits that hybridize to the region of interest, and then we can recover those hybrids using streptavidin-coated magnetic beads, washing away of target molecules. After analyzing the taxonomic profile of these 7 individuals, we identify a high abundance of Salmonella enterica, which is represented by the red color. And we map these breeds to the paratype C reference genome, and we were able to identify approximately 40,000 reads that map to this genome. And through the analysis of the amination pattern that is characteristic of ancient DNA, we confirm that this strain belonged to an ancient microorganism and is not associated to modern contamination. To further elucidate our finding, we performed an in-solution hybridization capture. We generated in-house butynylated RNA proofs transcripts from Salmonella typhimurium strain, and additionally, we sequenced at a higher depth. This strategy allows us to reconstruct a tenfold paratype C genome. So we decided to carry out preliminary phylogenetic analysis, including the Mexican previously reported genomes and 3 European ones. So we built a multi-sequence alignment of 18 modern genomes and 6 ancient genomes, and we performed a maximum latitude tree built with 70,000 SNPs. And according to this phylogenetic analysis, we observe a close relationship with the Mexican strains and paratype C modern genome. And as for the European ones, we observe a closer relationship with the pig-adapted Salmonella chlorosis strain. To sum up, we reconstruct parasite VC genome with tenfold X coverage from Mexico dated to the colonial period. We provide a direct evidence of this pathogen affected people elsewhere besides only the south of Mexico. Through the phylogenetic analysis, we determined a close relationship between our strain and the other two previously reported that was associated to Cocoliscli. We are interested in studying the pathogenic genetic variants that are related to the evolution and pathogenicity of Paratitis in Mexico during the colonial period. So I would like to thank to my supervisor and my lab mates, and especially to Dr. Ashley Begin, who has guided me in this type of analysis. So I'm very happy to answer your question. Thank you.